Okay, well, let's go ahead and get started. Welcome everybody for coming. This is Tools for Polyploids project. This is a computational session. The first one, there'll be two others. This first one, we want to do two things. One is for those who are not familiar with the Genotype calling software. What we ask you to do, if you have to catch up, you can look at the previous presentations, and then we'll be willing to and able to answer any questions that you have. So we had 4 presentations in the first workshop. One, the overview of genotype calling by Rulin, and he's actually on this call as well, so he'll be able to answer any questions. Polyrad by Lindsay, Fit Poly again by Rulin, and Supermassa by Marcelo. And so we'll go through these one by one and work from there. Now the people actually answering the questions beyond Rulin, who's one of the presenters, would be these 4. And actually Maria's not here, but Chris will be taking lead as we go forward. And Gabriel and Jeekin will be helping answer questions as well. So they'll take it as the questions come. And so I'm gonna go ahead and let Chris take over now, and I'll go in the background.
My slides is just, okay, let it in background is this one that we are talking about. So if you want questions, this is the time to make it them.
How do we compare the various ways of calling genotypes, and what are they good for, and what kind of data are they good for, and what kind of data do they not work for? How are they organized that way, and how do they compare in ability to call the genotypes?
How compare them?
Mm-hmm.
I think they all has different purpose, actually, like depend of the type of the data and what you are doing. And I think Marcelo talk, he mentioned which is the best point of for each one of them. And but I think, like, if you, in my point of view, because I have a bias, so if you build a linkage map and your linkage map is very good, it will tell you that the genotype calling is good. And but if you have like array data, and like Fit Poly and Supermassa can do good with both of them, and GBS data, Polyrad and Updog will also do a good job. So I don't know, it's a difficult question, Dave.
Yeah, I would agree with that. Indeed, there's of course the difference between read counts on one hand and array data on the other hand. Of course, if you have read counts, then you go to Updog, would be my first ID. And if you have array data, there are a few options: Fit Poly, Supermassa, Polyrad.
Last time we had a lot of problems with installation, like mainly about Supermassa. I don't know if people could install all the softwares. If not, we are here to help. We can share screen and try to help you if you had any problems with software installation too. I think we split the sessions, but if someone has questions about the other sessions, feel free to ask.
Can the audience give us an idea of which software they have used or are planning to use?
I have a question, a more general question about genotype calling. I'm just wondering whether genotype calling is still needed if you have a reference sequence and you know the position of all the SNPs. You can actually regress the phenotype on the allele counts, or say on the allele frequency, and map QTLs in that way. So we recently published a paper on this, and we were able to map QTLs that coincide with QTLs that were identified by classical linkage mapping. So I'm just wondering, it's a more general question, right? So if we can avoid genotype calling, if you have the reference sequence and the allele frequencies. So just I want to know your opinion about this.
My opinion would be that linkage maps are complementary to sequences. For instance, they tell you something about recombination frequencies, which are important also from a breeding point of view. I mean, you can, if you want to have a marker for a gene that is not going to be lose its linkage due to recombination very soon, well then you are interested in the centimorgan distances and not so much in the base pair distances, because but
the base pair distances you can get that knowledge from the reference sequence, right? So from a physical map. So I agree with you that if you have genetic distances in terms of centimorgan, it could be valuable, but still, so genotype calling is actually it is a process with a lot of errors. So if you can avoid that, maybe you're more precise in the location of the QTL without genotype calling.
I think it also very much depends on on the situation of the crop you're talking about. Indeed, if you have very well developed sequences, you are in one position, but there are also quite a number of crops where there is not very good sequence available, where you have to make do with sequences from related species.
I completely agree. So when you don't have a physical map, so you have to rely on linkage maps, right? But if you have a physical map, you can avoid that step of genotype calling.
Now, still, you want to know what is the genotype of your material. You are still interested to know whether your your accession or whatever your cross parent has your gene of interest in simplex or duplex. Well, because that makes a difference in what you can expect in the progeny, of course. So you need some way to assess the dosage.
Yeah, okay. So once you have identified the QTL, you can switch to other markers which are more, say, clear about the genotype than GBS, like kind of CASP markers, right? So then you can easily determine the dosage and determine the genotype of the offspring you're interested in. Yeah.
For developing a linkage map, I would say that dosage calls from arrays are much more safe, much more precise than those obtained from GBS.
Yeah, you're right. Yeah.
We have other question. I don't know if you finished. You want to ask?
Yeah, I'm finished. Yeah, I'm finished.
Okay. We have a question in the chat. Is if genotype calling is the same as SNP calling? And I understand that they are different, mostly when we are not talking about diploids. I understand that SNP calling is more for identifying the position of the SNPs. And if you're working with diploids and a single individual, yes, will be the same, because if you identify a polymorphism, it means that will be heterozygous. But in polyploids, you want to not only know about the position of the SNPs, but you also want to know the dosage. And if homozygous, if heterozygous, which dosage of the heterozygous, so it would be different procedures.
I see we have somebody working on bananas. How good are these softwares predicting dosage in triploids?
Well, again, so we recently published a paper on the mapping population being in triploid. And we skipped the genotype calling. So what we did was actually, so we sequenced, we had we GPS the progeny. They're all triploids. They were coming from a cross between a tetraploid and a diploid. So all the progeny was triploid. And we regressed the phenotype on the allele frequencies. And well, we identified two QTL, and we did a kind of classical QTL mapping using part of the markers that segregated as a kind of backcross in the progeny, and we identified the same QTLs. So that's how we want to proceed now in banana using triploids. Maybe, well, maybe this was a lucky shot, but yeah, so we could actually skip the genotype calling and still map QTLs.
Yeah, I know in rose breeding, they use triploids as parents as well.
Yeah,
And
so
I
but
guess that would be my question is if we use a triploid as a parent, and I presume you'd get more 1N and 2N pollen out of that, perhaps. How do you approach the genetic analysis of that situation?
no, so in banana we don't make crosses with triploids. So it's either diploid by diploid, or tetraploid by diploid to get triploid offspring. But we don't cross triploids with diploids or tetraploids. No, we don't do that.
So it'd be similar to, I guess, the watermelon breeding for triploids.
I'm not familiar with watermelon, sorry.
Well, we've also analyzed the Astromeria population, diploid times tetraploid. And there we did, we produced a linkage map, and that was based on fit-poly analysis. So fit-poly doesn't have any problems with ploidies different from 4. I mean, it does everything from diploids 2 to, well, of course, it gets more complex and less accurate when you go to high ploidy levels. But there's nothing special about the triploid to do those score linkage mapping. But we have never analyzed a population with a triploid parent in any crop.
No, neither did I. So
another challenge.
Yeah.
well, triploids are actually sterile, right? So you can't cross them.
Well, not in rose. There's some fertile,
Okay.
semi-fertile triploids.
And what's the ratio between 2N pollen and N pollen then in that case?
I fear it changes with the cross.
But it's, I mean, is it that one is very rare and the other is predominant, or is it more or less 50-50 of them? Or?
No, I think probably the diploid pollen is predominant, but not positive.
No, okay.
There's various conclusions from different crosses.
I guess you could split the population in a diploid part. What is it? What's the other parent? Is then it's diploid?
It could be tetraploid, it could be diploid. You can do it either way.
Yeah.
You get progeny out of it.
Anyway, you would have a progeny that's partly one ploidy and partly another ploidy.
Yeah.
Right?
Mm-hmm.
Partly diploid and partly either diploid or tetraploid. And I guess you could quite easily distinguish those two by some kind of clustering. And then you could do the dosage calling in the two parts separately.
Okay.
We have more questions in the chat. Can I say one?
Yeah.
About the GBS in allotetraploid, what pipeline for alignment of GBS sequence to the reference genome do you recommend? We did it through tassel GBS, and we are having problems mainly in the LED calculation and a very high number of heterozygotes. Yeah, maybe I can answer. We have other options for pipeline for doing this NIP calling other than tassel. That's I already have experience with Freebase and Jeekin. They can also be tested, but I would say to pay attention to the doses and also to the filters you are using, depending on the type of population. Maybe filtering the markers in the way that makes sense for your population is better than you keep trying lots of different SNP calling methods. You can too, they are different, but maybe your problem is more related with filtering than the software you are using.
Well, what kind of filtering are you referring to specifically?
Can be missing data. And also, when we are talking about GPS, we are expecting a lot of missing data.
Mhm.
If you have a mapping population, you can filter by segregation. That's very good filter. And if you are considering or not reads that align in several places or different reads that align in same place, this can do a lot of fake heterozygotes. So just pay attention of the quality that data that you are analyzing. So these are some examples. Other person asked if the Slack will be active after the meeting. Yes, we are always paying attention to the Slack, so feel free to post questions anytime. We have other question. Is I will read entirely. We work in Blueberry. We have a deployed F2 population generated from using two separated F1. We have genotyped this population and have millions of SNPs and high heterozygosity. The reads were aligned to tetraploid reference genome. My question to you, Roland, are one, can dosage needs to be counted? Two, can we use GWAS instead of developing a map? Kindly suggest a better software in this case. Thank you.
Well, yes, of course. Well, so you have a population, F2 population, obtained by crossing two heterozygous F1s. Is that what I understood?
What?
Yeah, well, so the ploidy level that you're talking about, is it diploid or tetraploid?
A diploid. We have
Diploid.
a diploid F2 population generated from using two separated F1. Yeah. But the reads, like it's a little tricky. Like the reads were aligned to a tetraploid reference genome.
Yeah. And the question is whether you could do a GWAS.
The first one is, can dosage needs to be counted? If it's deployed, I don't think so, no.
No.
And can we use GWAS instead of developing a map? Is the second question.
Yeah, well, there's for diploids, of course, there's loads of software available for both doing a linkage map and doing. Well, I think you don't really need a linkage map if you have millions of SNPs and you have them all aligned to a reference genome. That's also what Marnet was saying, of course, just now. So just in order to locate QTLs, I guess you could do a GWAS indeed.
Yeah, that's what we do. Yeah.
Yeah.
In Banana.
Yeah, and I can see here, like the question, he's saying F2, but I understand that this is outcrossing population, no? Because he says that's highly heterozygosity. Yeah. So you need to, if you want to build a map, you want to look for softwares that can deal with outcrossing population and not just inbred F2.
Yeah, well, of course, there's, for instance, Joinmap who can do that.
Mm-hmm.
Joinmap can make a map QTL. They can work with these populations, as far as I'm aware.
Mm-hmm.
I have never used an outcrossing population myself in this sense. But,
yeah, also OneMap. I work with OneMap, so if you have questions about how to build, I can help too. I don't know if I think we answer his question.
Yeah. And also, I think millions of SNPs is a bit of an overload if you have a population, well, of a few hundred individuals.
Yeah, he don't mention the number of individuals he has.
No, but well, unless it's a population that runs in the thousands and thousands.
Mm-hmm.
I mean, there's no point in a diploid to have million SNPs.
Mm-hmm. Yeah.
At least not for mapping.
So how many SNPs would you want in a diploid population? 4,000 or 5,000?
Yeah, maybe not even that. When you do QTL mapping in a biparental population, you can usually combine with, well, a few hundred SNPs. That's for the first rough mapping. and then you concentrate on a few target spots where your QTLs are, where you want to do high density mapping. And so that you generate extra markers, or you look for extra markers in that region. And you, well, what we then do in such cases is to self some heterozygous F2 individuals, heterozygous for the regions of interest. And, yeah, and just genotype the markers in that region, because it all, yeah, you cannot get a better resolution than the number of recommendations that you get.
Yeah.
And of course, if your population is limited to a few hundred individuals, I mean, yeah, there's just a few hundred recommendations in your population. So you can have millions of SNPs, but that won't make it more precise.
Yeah, the person include more information. The number of individuals is around 300 plants. The number of reads, I think, was millions. And the number of SNPs after filtering are around 100,000, of which 9% are heterozygous. Hence, can we use GWAS as developing map for QTL mapping? Could be difficult. I have other question here. Is about the TASO GPS and the difference of the performance of TASO for poly and TASO GPS, and if it would pay to realign with the TASO for poly. I think the only difference between TASO for poly and TASO GPS is the amount of reads they allow to align, because I think TASO GPS had a limit. If I'm not wrong, Gabriel, help me here.
Yes, yes, it's correct, Chris. I think the limit is 127 reads, the maximum, right? So TASO for poly allows for more reads. So if you're dealing with a polyploid population, it should be better, yeah. But I think you are dealing with allotetraploid, right? So it might be a good idea to first think like treat the data as a diploid species. So yeah, it depends. It all depends on the pipeline. I'm sorry, on the methodology that was applied, the expected mean depth that you have for each genomic region. Yeah.
Yeah, I would use the TASO for poly to not filter your depth, your reads, because as much you have of reads better for poly, most if we are talking about polyploids. So that's the difference.
Yeah, that's important. If you have the option, TASO for poly would be better. But I just mentioned to go for treat as a diploid before, because she says that she needs to pay for the analysis, I think.
Mm-hmm.
Mm-hmm.
Other question about the person that made a question about if they could use GWAS. So this is the continuation. My basic understanding is that for an inbred outbred population, you need to use QTL leakage mapping, whereas for germplasm, you need to use GWAS, as the LD will be high in biparental map population.
Yeah, if you ask me, indeed, I would say if you have made your own cross population, I would use software that is designed to analyze these populations. Like I mentioned, JoinMap and MapQTL. These are not free software, by the way. They are commercial. And indeed, GWAS is not designed for analyzing a biparental population. That's for sure. But yeah, you are probably able. You will find the QTLs anyway, also with a GWAS. But of course, yeah, the linkage blocks are much larger than you would have if you have just a panel of more or less unrelated accessions.
In the typical GWAS paper, you always see 1 or 2 or 3 markers, whereas if you use an F2 population, you run a GWAS, you're going to see your peak. You're going to see the most significant markers, and then you're going to see a fall off of markers because LD blocks are so large.
Yeah.
So you can still pick up the QTL and it's still valid at least for the very strong effect QTL. You're not going to pick up some of the smaller effect QTL, which will need linkage mapping and other types of a QTL, actual QTL mapping. So that's what we've seen in roses.
But in GWAS, you also can fix major QTLs to find minor QTLs.
Thanks. However, due to high heterozygosity, developing a correct linkage map could be difficult. Hence, I have used GWAS approach, but will it be questioned when reported in a paper? I don't think a high heterozygosity will be a problem if you use these softwares that are ready to deal with this type of population. Use JoinMap or OneMap, that will be fine.
Or even Map Poly and PolyMapper, they still can map diploids. We've done it before.
The softwares built for polyploids can also build for diploids. Yeah. So if you prefer, you can also use them. Then how should we select polymorphic markers? Those polymorphic between F1s, parents, or those between grandparents? You can, like if you see the tutorials, we have, I think both softwares, JoinMap and OneMap has a way to codify each type of markers depending on the genotype of the parents. So you just need to, and we have like types and codes for each one of the types, and you can use all of them depending on the parents' genotypes. I will include the both softwares tutorials in the chat. If someone can help me, I will catch the link. The question is, the person will look for the manuals, but yes, if you have more questions, just let us know. You can ask about these two softwares too in the Slack or send us email. Okay, I will just change the emails be available here. This is our mails.
So if nobody's having like theoretical questions, are people having problems installing?
Ryan is asking, I will just open quote. I am exploring the presentation and packages and appreciate the work all have done and presented on. Not on top of genotype calling, but any recommendations on our package for sorting out parentage individuals in a polyploid polycross with microsatellite data. I think the Amadeo package can deal with this, no? The AGAGA metrics.
I thought Jeff Edelman had a package that could deal with that. I don't know the name of it.
Yeah, you can do parentage analysis, but I think it's for SNP data. I'm not sure what this is.
Not microsatellites. Okay.
Yeah.
So what kind of sorting out parentage are we talking about? Like, is there like an F2 that you don't know what the correct parents are or something like that?
And what crop we dealing with? With polycross? Mint.
Mint mixed population.
And how many SSRs?
May not have enough markers to do this right.
Ryan, when you say a mixed population, I'm not quite sure I understand what that means. Do you have a specified number of parents that you interplanted to collect seed from? And then you want to decide which of the combinations is most likely? Or is it a lot broader than that?
And do you know the mother plants and are looking for the father only, or do you know and don't you know any of the parents?
Good question.
Yeah, so it seems like he wants to perform a paternity test, right?
Mm-hmm.
Yeah, but I don't know if there's any package available that does this like in a seamless way. I'm not sure about that. Do you know any package? Chris, Jeekin, Roland?
So I mentioned the Amadeo package before, but it's not able to do that.
Yeah.
But with only 10 microsatellites, and if you don't have a lot of potential fathers, you can probably do it by hand relatively easily.
Yeah, that's my thought also.
Yes. Mhm.
but it would be tedious.
You can program it maybe. A few lines of R code should do it, I think.
Well, that's a project you can do, Roland. Just 2 lines, simple.
2 lines is a bit small, but it shouldn't take a whole day, I would say. So how many fathers are there? How many potential fathers? You can do that by hand.
So how possible it would be depends on how variable those microsatellites are now and how different
Yeah,
the parents are. But yeah, but 2 codes of R would be nice too. Hint, hint. So has anyone had any problems with the installation of any of the software that you've wanted to use? Okay, Chris, you have any thoughts?
And I don't know.
I'm
I
sure
think we did
Gabriel
a
wants to say something, right?
I think we did a good work in Slack last year, so installation now is easy.
Yeah. Yeah. I need to assume that I was expecting lots of people with problems with Supermassa, especially because we had many problems last year, last workshop. I'm sorry.
Yeah, the person is asking for a list of software and that do linkage and key-tail analysis in Polyploids to try to install and everything. You can find this list in the website, in the Polyploids 2 website. I will ask someone to send the link.
Yeah,
all
I will
software
send it.
presents there. Okay, thank you. So you can access there. I don't know if the key-tail poly was already updated, you know, Gabriel?
No, it's not updated yet, but I messaged Delaney, so yeah, it should be updated soon.
Hi, I have a problem. Yes, we have a problem. I have a problem with installing package GWAS poly. Okay. Do you have an error message or something?
So I will share my screen.
Okay.
to-
I will stop here. I can see the problem already. The GWAS poly is only in GitHub, so you will not be able to install through this path. You need to run the install GitHub line. I think you
okay.
cannot.
So I have to go.
I can put the commands in the chat.
So options responsible.
Okay. Yes, just.
Okay, thank you so much. I saw that.
Yeah, let us know if you could install that.
Sure. Let me first stop sharing my screen. Thank you.
Welcome.
Anybody else having trouble with installation? Well, it's great to hear the installation went so smoothly.
Yes.
If this session is easy like this, the other ones, because the other ones are all in R, so
Yeah.
we would expect more problems today.
Yeah.
Should expect some
Actually-
problems in the workshop as well.
Yes, that's true. And actually, the mapping session, we may have some problems with poly origin, maybe. I'm not sure because it's not in R.
Other question.
I see on the chat, link for fit poly in your software link comes up with an error message from Susan. So do we have a wrong link? It says object
Yeah.
not found.
Yeah, there is an error in the link in the website. Yeah, thanks for letting us know. We will update it. The correct link, I will send the correct link in the chat.
Yeah, send it to Delaney too, so she can correct it.
There's other question. I have a follow-up question. Do you mean that all the software need to be run through GitHub only, or can they be installed in RStudio? No, the problem is that the software to be hosted in only GitHub. Usually, when it's R package, they are usually in CRAN. That's the main repository of R. But some software are only in GitHub. And then what you need to do is, instead of installing from CRAN, you install from GitHub. And the DevTools package help you to do this. So will be installed in your local RStudio, but the link is to GitHub and not CRAN. That was the error that the other person was having.
Again, in chat for GWAS Poly, it shows this error. Enter one or more numbers or an empty line to skip updates. Library GWAS Poly.
Sometimes when you're installing a package, the RStudio asks if you want to update the other package. But instead, because probably you select all the lines and run at once, it's instead of giving the answer, you gave the next line. So if you run again, just the line by line, you should be able to answer the question with a number. Let us know too if you have problems with our language in general. We can help too.
I guess if there's no other questions, remember that the Slack channel is available. You have everybody's emails. They are more than willing to help you with all your issues of installing and understanding what's going on, because these programs are fairly complex. Any last comments, Jeekin, Gabriel, Chris, Rulin?
One other question here.
Okay, good.
So we have to install the software from GitHub and are using DevTools as given in the links. Yes, using DevTools, you can just include the link that we give you from the repository in GitHub, and the RStudio will install will install the package for you. So some of the softwares has two versions, one in CRAN and other in GitHub, and they consider the CRAN version the stable one, and the GitHub version the under development one. And sometimes you will prefer to use the most updated instead of the stable. So even if it exists in CRAN, sometimes you want to install the GitHub version. Yeah, the same person sent me an image about KTL Poly, and the KTL Poly now is on CRAN too. You can install package KTL Poly, or you, Gabriel, can you send the link of the new version of KTL Poly?
Yes. I put the link on the chat.
Okay. We will update the Polyploid Tools website with this information.
It's much easier to install now. Very easy.
Thanks to Gabriel.
Yes. I'm putting the link here in the chat.
Thank you for answering all my questions. We are new to these programs and polyploid crops. Hence, may I request you to assist in further installations? Thank you. Yes, we will be available on. And if you will try this week to install, we have other sessions, so you can ask more questions in the next sessions or in this line.
Or if they need help, they can share their screen right now, because I think that's what we're here for this session anyways. Installation of any of these linkage calling, the Genotype calling things, or linkage software. I guess we could also help them with that, so it doesn't take up any of the other times. Because I feel like the linkage mapping and QTL mapping, there's a lot of conceptual things that might be asked too. So whoever just asked if they need help getting things installed, would they like to tell us what they need help on?
I just put back here our emails and the Slack address. So if you want to ask question later, too, feel free.
Well, I guess if there's no more questions, we can end the session and we'll have another session next week. So last chance for a question. Okay, with that, let's go ahead and end the session. And I appreciate everybody who came and our computational team for answering all the questions. And of course, Rulon for being with us to help us with some of the other questions. So make sure you have the Slack channel for your questions between sessions. and computational team will answer those.
Okay, thank you, everyone.
Okay, thank you.
Thank you
Bye-bye.
very much.